Okay, so let us get started for today.
Hello.
Hello Subha.
Hi.
Okay, so we'll get right into it. Hope my screen is visible.
Yes, it is visible.
Okay, so welcome to the week 8 of Introduction to Developmental Biology. This is, as you would know, a PMRF-NPTEL session in which we do problem solving sessions by solving the previous year's quizzes of the same course to get a better understanding of the concepts behind the entire course, as well as how to answer questions in the course of this year as well as the final exam for NPTEL. A quick introduction from my side. I am Anuvab Chatterjee. I am a PMRF fellow at IIT Kanpur and I will be taking this course. And how we do this course is that I will show you a question from the previous year's run of the course and we will try to solve it together, try and answer it, and then I will show you the concepts behind it, and that will help us answer the question finally. Okay, so this week we are dealing with further with body plan specification in Drosophila and learning more about different types of genes that are expressed, that are firstly bicoid nanos, followed by the gap genes, the pair rule genes, the segment polarity genes, and we'll end up with the homeotic or the Hox genes. And by this time, we have already shown in the previous week's course how exactly the initial asymmetry, the anterior posterior, the dorsal ventral are formed in case of Drosophila embryos, how the bicoid and the nanos gradients are set up by the gherkin and the pipe. We have already covered it. Now we will go forward with it. So the first question that we will tackle today is, this is an experiment that we dealt with in the course's lectures, and this the question is based on this. So select the correct conclusions drawn from the ligature experiment on the smita, smishia. The options are: bicoid and nanos are involved one at a time in the Drosophila development. There must be two decision points, one at the anterior, one at the posterior of the embryo. Fushitarazu determined the gradient of genes expressed, and engrailed was seen only at the posterior end of the embryo. Yes, Shobha?
I cannot see the screen. The I cannot see the screen
Ah,
that you
okay,
show.
okay, 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 okay. Let me. Oh, my PPT is visible?
And now it's visible.
Yes. Okay, perfect. So, yes, repeating the question, this is the first one that we tackled today. It is select the correct conclusions drawn from the ligature experiment on the mixed media. The options are: bicoid and nanos are involved one at a time in Drosophila development. There must be two decision points, one at the anterior, one at the posterior of the embryo. Fushitarazu determined the gradient of genes expressed, and engrailed was seen only at the posterior end of the embryo. So if you have gone through the lectures, this I think you can try and answer this. So the options are A, B, C, D. You can choose whichever you think is the correct option. Okay, so the option Subha has chosen is B. There must be two decision points, one at the anterior, one at the posterior of the embryo, which is correct, but we will still go through what exactly the experiment is. So by this time, people were still trying to figure out how exactly the body plan is established in insects. So there is this person, scientist Klaus Sanders, who instead of working with Drosophila was working with another segmented insect known as Smycia. So what he did is that he went ahead and took the embryo of the this particular insect and tied a ligature, which is essentially a string in the middle of the entire embryo. So a ligature in the middle, so essentially somewhere here in the embryo. And what interestingly he saw is that the anterior part, so the head structures and the upper thoracic segments were formed. The posterior half, the tail segments and the lower abdominal segments were formed, but the middle segments, none of them were properly formed. Now he repeated the same experiment at a later stage. So he allowed the embryo to go further and then did the same thing, tied a ligature around the middle of the embryo. And this again reduced the number of missing middle segments. So further of the body was correctly patterned. You have more, you have the head and more of the upper thoracic and the tail and the more of the lower abdominal segments, but some segments in the middle were not properly established. So obviously Klaus Sanders knew how morphogens work, so he figured out that there must be a morphogen which is emanating from the head or the anterior most point and a more different morphogen from the posterior, both of which are determining the body plan along the anterior posterior axis. And when you tie a ligature marks, these morphogens cannot drift further after the ligature. So the effect of the anterior morphogen is not seen towards the posterior, and the posterior morphogen is not seen towards the anterior, which is important for the middle segments to form. So these observations suggested the existence of a morphogenetic gradient emanating from the two poles, the anterior and the posterior. So we will look at how the embryos look. So here is a Smithia embryo with normal head segments and the like normal body plan. So you see there is this segmented pattern all the way throughout the body. Here's the head, here's the tail. But once you tie a ligature through it, the middle segments are not properly formed. So this is what he determined, that there is a particular morphogen here and a particular morphogen here, both which are coming from both the ends and both are required to form the entire body plan. So so now currently we know what the two morphogens are. The anterior morphogen is bicoid, which is deposited at the anterior pole, and here is a RNA in situ hybridization for bicoid, so you can see it is completely in the anterior pole. And here is the posterior morphogen that he predicted, which is nanos, which is also, as you can see, is in the posterior pole. And here is nanos protein, which is further along because the protein can diffuse, whereas the mRNA is stuck here. So the answer to the question, what are the conclusions? The conclusions are, there are two decision points, one at the anterior and one at the posterior of the embryo, which helps in patterning the entire embryo. Okay, so that one was easy. We move on. Okay, so we have understood that yes, there is a bicoid uh, emanating center at the anterior pole, a nano center at the posterior pole, but how exactly are they maintained at the anterior and the posterior? So we ask the question, which of the following steps are involved in determining the position of the nanos mRNA? So the options are, initially the nerve cells deposit nanos mRNA at the anterior end of the oocyte, which then diffuses throughout the oocyte cytoplasm. The nanos mRNA is carried to the posterior end on motor proteins like the bicoid transport. At the at the posterior, Oscar anchors and thus concentrates the nanos mRNA, and Oscar dissociates the nanos mRNA from the SMAU complex at the posterior end. So this is a more than one correct option kind of a question. So you can choose A, B, C, D, multiple of them. So I'll give you a second to try and figure this out. Okay, so we got an answer which is A and C. Initially, the nerve cells deposit nanomRNA at the anterior end of the oocyte, which diffuses into the rest of the oocyte cytoplasm, and C, at the posterior, Oscar anchors and thus concentrates the nanomRNA. Both are true, but let us look at it. So this is how diffusion and anchoring of the nanomRNA occurs. So the nanomRNA is maternally deposited. So by this time, there has not been fertilization done yet. So it is just the oocyte nucleus, which is not transcriptionally active. So it is not making anything. The nerve cells, which are on this part, they pump a lot of proteins into the oocyte. One of them being the nanomRNA. The nanomRNA gets into the oocyte and is essentially diffusing throughout the cytoplasm. But the Oscar is another mRNA which is deposited by the nerve cells. It actually hitches a ride on the cytoskeleton network in the entire oocyte and is first concentrated at the posterior end. Next, what Oscar does is that it catches hold of all the different nanos that is floating around and concentrates it on the posterior. But another thing occurs. Our nanos mRNA is bound by another protein which is known as SMAUG. This protein, whenever it finds nanos mRNA, it binds to the 3' UTR and doesn't let all these other nanos mRNA which are diffusing freely throughout the cytoplasm to start translating into the nanos protein to do its job. This SMAUG protein is actually removed only in the nanos mRNA that are bound to these posterior Oscar proteins. So nanos is only translated right here, so that you have a particular center of nanos right here where the protein is being made, and none of the other no other place in the entire cytoplasm is producing nanos. So three things occurs. The first, the nerve cells deposit the nanos mRNA and it diffuses throughout the cytoplasm. Oscar anchors the nanos mRNA to the posterior end, and Oscar dissociates the nanos mRNA from the SMAUG complex so that it can actually be translated into nanos protein only at the posterior end. So we have a posterior pole of nanos and not nanos being in the entire cytoplasm being translated. Okay? Okay, so that was that. Yes, Suba?
No, I raised my hand to say okay.
Okay, perfect.
Sorry.
Okay, so we move on. Actually, getting to the major thing that we are going to deal with today. And we started off with a question: which of this is not a gap gene? The options are empty spiracles, orthodentical, cruple, and bicoid. And Subha has already said it is bicoid, which is not a gap gene, which is correct. Bicoid is part of the two anterior and posterior molecules that initially create the asymmetry, and all of these are the gap genes. But let us actually go through the entire concept of what is actually going on. So after the initial anterior posterior asymmetry is established by Gherkin, Torpedo, and the entire things that we have discussed in the previous week's course, we have bicoid in the anterior, a bicoid mRNA in the anterior, and nanos mRNA in the posterior. Once now, as fertilization has occurred and the nuclei actually now starts replicating. So what Drosophila does is that instead of the cells dividing and forming two complete cells, it does not undergo cytokinesis. It only undergoes karyokinesis. That is, there is a huge cell which has multiple nuclei in it, and once these nuclei are formed, they travel to the cortex, or you can imagine this being a huge ball or an oval ball, and the nuclei are deposited just under the surface of the ball. There is nothing in the middle, so this part is empty, but all the nuclei are at the surface of the entire oocyte. But this is not no longer a oocyte, this is an embryo now, which has already been fertilized and nuclear division has formed. So there are no cell boundaries, there are just multiple nuclei inside one cell boundary. So this is the syncytial development that we talk about a lot. So a lot of nuclei which are in a syncytium, that is a lot of things being in the same place and not being divided in boundaries. So since the nuclei are like this, these nuclei can be affected by the same cytoplasmic components, which are the bicoid protein and the nanose protein, which are being translated at the moment. And you can see there is this gradient that is present. So a red representing the bicoid, which is maximum at the anterior and diffuses, and the blue being the nanos, being maximum at the posterior and diffuses at the other end. But this is just two proteins or two genes that are asymmetrically divided in the entire embryo. But from here, how will you actually form the entire body plan? So the thing is that only bicoid and nanos do not form the entire body plan. They are, you can imagine them being the primary players, which then activate secondary players or secondary molecular players. Then they activate the tertiary molecular players, which one by one actually make sure that only some of the nuclei here actually form the head region, some nuclei here from the thoracic region, some here from the abdominal region, some here the tail region, and then specific tissues originating from these regions. So bicoid and nanos are just the initial players that kick off the asymmetry, and they will now start uh, ex- uh, making sure that these nuclei spatially are expressing different proteins or genes so that they form different regions. So after cellularization, which is essentially the nuclei actually now undergoing cytokinesis and forming different cells, this is what the entire embryo will look like. So by this time already the head region, the acron or the head cap, the anterior most region, the head region, the thorax, the abdomen, the telson or the tail region, and the pole cells are already determined. So our entire idea is how does this gradient of bicoid and nanos determine which particular region will form which particular type of tissue. So we go a bit back and figure out how exactly people figured this out. So in a normal wild type embryo, where we have bicoid at the anterior region, we form the normal wild type Drosophila body plan, which has acron, head, the thorax, the abdomen, and the tail. Very good. But when people were looking at bicoid deficient mutants, so there is no bicoid here because there is no bicoid anywhere, then they actually did not form a head here. They formed a telson, a abdomen, and a telson. So essentially, none of the anterior structures were formed. Next, yes, Subha.
Yeah, I'm wondering, if the embryo has only the telson abdomen and the telson, the tail region,
Mm-hmm.
so these don't develop further, right?
No,
Because they sleep.
these are embryonic lethal. So this is essentially how they figured out. So they had a lot of embryos on plates, and they only looked for embryos which are dead, and then they picked them up and then figured out the morphology of them. So yes, these are
Okay,
embryonic lethals.
okay,
Yeah. So that is the first experiment they did. Next, they figured out, okay, so bicoid, if bicoid is not there, then we the body plan is all messed up. So how about we add bicoid? But interestingly, they added it to different parts of the embryo. When they added it to the anterior most region, the body plan was actually rescued. You again have acron, head, thorax, abdomen, and telson. But interestingly, when they added it to the middle of the embryo, the head region started forming in the middle. So this is essentially, we have already learned that this is how morphogens act. Wherever there is a highest concentration of bicoid, there the tissues will take that particular fate. So highest concentration of bicoid has the head fate, and that is why even if you inject it into the middle, the head forms in the middle. And finally, they also tried it out with a wild type embryo which already has bicoid at the anterior pole, and they added bicoid mRNA to the posterior pole as well. When they added it, they saw that none of the posterior components formed. It was essentially a duplication of the anterior. So you have acron head thorax followed by the normal acron head thorax coming from the other end. So a two-headed embryo. So they figured out, yes, the becoid helps in forming the anterior body plan, and that is important. So now they started looking at how it does it. So this is, people have figured it out, so we are just going to go through it easily. So we have bicoid here, we have nanos here. They actually first, like as I said, you can imagine this is the primary asymmetric genes. This give rise to a secondary set of genes which cause a localized asymmetries. These are known as the gap genes. So there are different gap genes, and they are expressed, they are essentially 6 to 9 depending on what you are, well, like who you ask. But the thorax has essentially four important ones that we will look at, and they appear as different stripes, like huge stripes that we see right here. So they and these are called gap genes because when you do not have one, you have a mutant of one of these particular genes, then the part where the gap gene was being expressed, that part does not occur in the final embryo. So obviously you imagine there being gaps, so you imagine there, this particular gene, if it is not there, this entire thing will not be there, so you have a gap in the embryo, and that is why they were named gap genes. Next, these gap genes in turn activate a tertiary or a third round of genes which are known as pair rule genes. Pair rule genes are interesting because they actually form this cool stripe like patterns, which we will also look at, in which the genes are expressed. So this is the same gene which is expressed as a stripe throughout the embryo, and these stripes are actually the parasegments which will help determine different parts of the final adult fly. And these pair rule genes again help in formation of another set of genes which are known as the segment polarity genes. So once the parasegments are determined, so parasegments are so Drosophila has a segmented body plan. So it has different segments, which I will actually show it to you, then it will be easier. Ta, 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 ta. Yes, this image. So Drosophila has a segmented body plan. So it has a T1 segment. So this is the thoracic only we are talking about. It has a T1 segment, a T2 segment, a T3, A1, 2, 3, 4, 5, 6, 7, and 8. So different parts of the body forms different structures, and these are very well spatially defined. You can look at the insect and see that there are these segments. So these are also present on the embryo, and here is where they are actually determined. As you can see, this is the T1, the T2, the T3, and then following we go on all the segments. So these are actually determined at the embryonic stage. So our pair rule genes are expressed in particular locations which are known as the parasegments, which do not align with these well spatially differentiated boundaries, but they appear between them. So on the boundaries of them. So part here, part here. These are where the pair rule genes are expressed, like a stripe right here on the boundary between two segments. And the next set is known as the segment polarity genes. The segment polarity genes are expressed only at the anterior of some of the segments, and other genes are expressed on the posterior of the some of the segments. So essentially, as we have already determined the anterior and posterior of the entire embryo, now the segment polarity genes determine the anterior and posterior of the specific segment. Because you can imagine that if you take a look at the T2 segment, only the anterior must form the wings and the posterior must form the legs. So obviously in this small part of the body there is also anterior posterior different. There is a difference between the anterior and posterior, and this is where the segment polarity genes come into play. They determine that yes, this is the anterior, this is what will form the wings, and this is the posterior that will form the legs. So we will go slowly through how everything happens, but essentially you have to remember that these are the different set of genes. The gap genes follow, which in turn help in expression of the pair rule genes, which help in expression of the segment polarity genes, and all of these form the homeotic genes, and these are like the most important regulators of body planning. These genes are expressed in specific locations throughout the entire body, and they will determine where what will form. So we look at examples where if you take a homeotic gene from where it should have formed our legs, the Drosophila's legs, and put it into the region where it should have formed the antennas, then the legs will form in place where the antennas were. So essentially, they determine where what structure should form. So similarly, there are genes which are known as the PAX genes, which determine even in vertebrates where the eyes will form. So if you take the PAX gene somewhere and put it somewhere else, you will have an eye forming out of nowhere there. So the homeotic genes are like the most powerful regulators of body planning, and all of these genes essentially control where exactly the homeotic genes will function. But we will go through them very slowly so we get an understanding. Okay, so we go to the first step. So we have bicoid, we have nanos. You can see that the bicoid has a gradient maximum at the anterior region, diffusing and slowing down as we go to the middle. So this bicoid and nanos gradient give rise to the first set of genes, the gap genes. As you can see, the gap genes have these, they span a large region, and this is actually a in situ hybridization. So this shows which nuclei are expressing which gap gene. So at the highest concentration of bicoid, and you have to remember bicoid and nanos both are transcription and translation regulators. So bicoid helps in transcription of the gap gene hunchback. So wherever there is a very high concentration of bicoid, only there will be a expression of hunchback, followed by the expression of Kruppel. And as you can see, the gap genes can overlap, and you have, if you have hunchback in red, Kruppel in green, then you have the overlap shown here in yellow. So this is how the gap genes function. So they are expressed in huge patches along the entire anterior posterior axis. Next we have the pair rule genes. As I said, they have this very nice stripe pattern that are expressed in the parasegment. So you have a stripe of Fushi-Tarazu throughout the entire embryo like this. And finally, the segment polarity genes, which will be expressed for in case of engrailed is expressed at the anterior portion of each of the segment, determining that yes, this is a segment, this must be the anterior portion of the segment where anterior specific parts will form. So these are all the genes, and we are going to talk about, yes, Suba.
So in the segment polarity gene, you mentioned the engrailed. So at this stage, they are forming a larvae. They are turning into a larvae, isn't it?
Yes, so
Because it's like an embryo. This is like a one cell, but at this point, they are out of the syncytium, and they are cellularized, and this is now turning into a larvae,
yes,
or because
so
the segmented looks like it's like a larvae, so that's why I'm asking.
yeah, that is a
Yeah.
good question. Let us just go through this. So, what happened when we say a larvae? That we imagine that the embryo has actually been born. So, larvae is the first stage after the egg, like the, like you can imagine when the, it is an embryo
Oh,
whenever it is inside the eggshell. So, what you are talking about is a
okay.
developed embryo. It is still an embryo though. Okay.
It's still embryo. Okay.
Yeah, and you have raised a good point. So when we are talking about the gap genes, it is still a syncytium. When we are talking about the pair rule genes, it is still a syncytium. But by the time the segment polarity genes come up, the embryo has cellularized. So the cells are actually divided into smaller parts. Instead of there being a complete cytoplasm with lot of nuclei in them, now they have different cells. But it is still a developed late stage embryo.
Okay.
Yeah, like this gap gene, you can actually still see the nuclei divided, but by the time we have the segment polarity genes, the cellularization has already formed occurred. So this looks like a larvae because it is almost developed. But you are correct, yes.
Okay.
Yeah. So yes, so we go all the way back and we think about it a bit and we figure out how exactly bicoid and nanos gradient will form these gap genes first. So we go step by step. So we know that bicoid, highest amount of bicoid is here, highest amount of nanos is here. The first thing that occurs is that bicoid at a very high concentration starts the expression of the first gap gene, which is giant. So we have a huge portion of giant right here, and a low amount of uh, a low amount of bicoid is necessary. Yes, Subha?
So earlier you mentioned that the bicoid helps in translation of the hunchback,
Yes,
right?
yes.
But how did the giant is there in the anterior
So
region of where the bicoid is localized?
so as I said, there are what do you call it, overlaps between the expression. So here,
Okay.
even in this image, since we have do not have giant, you would have seen giant right here.
Okay.
I-
Okay. Okay. Okay.
And in this image, you can see that giant is here. It has just overlapped with hunchback.
Okay. Okay.
Yeah. Okay. So we have giant here at the anterior most pole, followed by hunchback. So even a low amount of hunchback, low amount of bicoid can start the expression hunchback. So we have giant, followed by hunchback, and next we have crupple. So here, interestingly, the gap genes, they actually hate each other, but they hate the alternate ones. So giant doesn't like crupple, crupple doesn't like giant. So you can see right here. So giant opposes crupple coming up right close to it. So there is going to be a region where there is no crupple and only hunchback here, because giant here says I do not like crupple, so I don't allow it to be expressed here. Crupple says yes, I also do not like you. So there is this region in the middle when neither giant or crupple is expressed and only hunchback is expressed. So you have this giant, hunchback, and crupple. Similarly, on the other side, the same thing happens. Hunchback, it uh, actually inhibits giant at the posterior end. So where there is hunchback on the posterior end, there is no giant. Then we have giant. Giant does not like nerves, which is the next gene. So there is no overlap between nerves and giant. So here there is a huge percentage, there is only giant, followed by crupple. So this actually helps us understand this better. So hunchback, wherever it is expressed, it does not like giant. So hunchback has a separate domain, no giant there, which we see right here. Giant does not like nerves, so we have giant being expressed where there is no nerves, followed by domain of nerves, and finally we have crupple, which also nerves doesn't like. So we do not have much of an overlap between them. We have crupple here, and then so essentially this shows us that each of the genes have a specially dedicated domain in which they express, and since they do not like each other, they make sure that domain only expresses them. So nerves doesn't like crupple, so only nerves is expressed here, even though if nerves was not there, crupple's expression domain which have increased throughout entire region. Make sense? So that is why we have this huge patch like expression of the gap genes. Does that make sense? No.
Okay. Yes.
Yeah. So essentially
Yes.
everyone hates each other, and that's why we have a patch like expression, and very specific patch like expression. Only at the boundaries is where we will see a overlap. Here we have so this is just a table to show you different gap genes. Actually not this one, this one is the one we want. So in the trunk we have the more important ones: hunchback, crupple, nerves, and giant. At the ends we have two important ones which are tailless, and the other one is buttonhead. Tailless and buttonhead, both of them determine the extreme head and tail regions, and Huckelbein orthodentical and empty spiracles actually help in the small, like not the anterior most, but the head region in the case of the orthodentical and the empty spiracles, and Huckelbein helps in the tail, like the not the most posterior region but just posterior. So this is essentially an entire table of it. But after all that discussion, the question essentially was which of the following is not a gap gene, and bicoid is not a gap gene, which was correctly answered. So from this entire discussion, I just want you to know that bicoid and nanos, due to their gradient, and since these are morphogens, so higher concentration will for lead to expression of one particular gene, medium or moderate concentration to a second gene, and less concentration to a third gene. That is how initially the gap genes have started to express. Next, these gap genes actually regulate each other and actually down regulate each other, so that where one of these gap genes are expressed, the other is not. So we have this patch like expression. If they didn't down regulate each other, or if they were not present in that region, because of the concentration gradient, the domains of each of the gap genes will increase. So there are experiments in which they have removed giant, and nerves has increased its entire domain to this huge place, instead of being spatially determined to be just in the middle stripe. So gap genes, huge patches which are spatially separated, and they have an overlap in the middle. So these are gap genes. Okay, moving on to the next question. Mutations in blank genes cause loss of portions of every other segment, whereas mutations in blank genes cause defects in every segment. So one of these cause defects in every other segment. So you skip one segment, and defects in these one cause problems in every segment. So the options are essentially segment polarity and pair rule and gap being jumbled up. Okay, Subha has said that mutations in pair rule genes cause loss of portions every other segment, and the segment polarity does it in every segment, which is absolutely correct. Let us look at it now. So the pair rule genes come as the next rung down the ladder. So pair rule genes are expressed in every other segment. So essentially they are expressed in this nice stripe-like patterns that we can see right here in case of Fushitarazu, and these are expressed not in the segments, as I said, at the boundary of the segments, the para segments. So they essentially are formed at the boundary right here. We see is where they are formed. And if pair rule genes are mutated, then these boundaries of every other segment is affected. So you have half the or the alternate segments being affected in case of the pair rule gene mutations. But in case of segment polarity genes, these are expressed in every one of the segments, but at either the anterior end or the posterior end. Engrailed is expressed at the anterior end of each of the segments, and so when you have a mutation, this anterior end of each of the segment is not there in the final embryo. So with the segment polarity genes, we have problems in all the segments. And here we have an example of two in situ hybridization for two pair rule genes. One of them is even skipped, one is Fushitarazu, and as you can see, even skipped is in stripes which are not overlapped by Fushitarazu. So essentially they are alternate, red, black, red, black, red, black, because they are expressed in alternate parasegments. So these two parasegments are side by sides, and they are not expressed in each but in alternate ones. And that is why you have a zebra crossing kind of a pattern when you do it for two different pair rule genes. Okay, so mutations in pair rule genes cause loss of portions in every other segment because they are expressed in alternate segments, and in segment polarity genes cause defect in every segment, individual segments, because they are expressed in every segment. Okay. Okay, so perfect. Moving on. Which of the following is or are not activated by bicoid? The options are hunchback, caudal, nerves, or giant. So A, B, C, D, whichever you think is the correct option. Okay, caudal and nerves, we got the answer. So yeah, let us look at it. So we are so as hunchback being a gap gene is helps in formation of the anterior region. There is caudal, which is another gene which helps in the posterior region. So initially, as we know, bicoid and nanos mRNAs are very narrowly localized at the anterior and the posterior respectively. But hunchback and caudal are diffusing throughout the entire cytoplasm. So they can, if they were allowed, they will start translating anywhere. But bicoid actually inhibits caudal, which is nanos activates caudal. So this is the posterior defining gene. So only it is expressed in the posterior. Similarly, hunchback is the anterior gene, which is activated by bicoid and inhibited by nanos. So it is only present in the anterior part. So here we have a flowchart for the same. Bicoid protein inhibits caudal, so only caudal is in the posterior. Nanos inhibits hunchback, so hunchback is in the anterior. So we have this sloping kind of a thing. So the answer is caudal. So nerves is a gap gene which is also activated by bicoid. So we go back. Yes, nerves is also activated by bicoid, but a low concentration of bicoid. But the posterior part is where caudal is present, which is inhibited by bicoid, and that is why the posterior regions stay posterior and do not come into the anterior. Okay, so the answer, which of them is not activated by bicoid, it is caudal, which it actively inhibits. So it is a transcriptional inhibitor of caudal. Okay, so same we have the same graph again. So in early cleavage embryo we have the same graph, and now we have to choose A, B, C and D. Which of these are the following? So we essentially, it is a match the column, but they have already matched the columns for you, so you just have to choose whichever is the correct option. Okay, so Suba is saying it is C. So A is hunchback, yes. B is okay. Let us look at it. Let us look at it. Okay, so as we have seen that bicoid and nanos mRNAs are very spatially restricted, so they have a very sharp gradient. They fall down very fast, whereas bicoid and nanos have a mild, shallower gradient, so they fall down at a much lesser slope. And caudal and hunchback, they follow suit, but they always have a higher concentration. So this is how it looks like. So bicoid and nanos are the shallower gradients, the red and blue. Green and black are caudal. So we look at it. So the green is hunchback, The B should be, okay, I was looking at it wrong. C option is this one. Okay, yeah, it is a hunchback, caudal, dicoid, and nanos. Yes, C is the correct option. So this is ABCD. This is also ABCD. Excellent question formation here. Okay, so C is the correct option. Okay, moving on. Gap genes are regulated by mutual antagonism among non-adjacent pairs of genes. So we have seen hunchback doesn't like giant, doesn't like cripple doesn't like nerves, and that is why we have separate spatially distinct patches of the gap genes. But what regulates the formation of pair rule genes being in these very nice stripes? The options are promoter, methylation pattern, histone modification, and enhancer modules. And Subha has also already said that it is enhancer modules, which is the correct option. But let us look at it. So this is the same image we have looked at, the red and black being two pair rule genes, even skipped and Fushitarazu. And we see that we have very nice stripe-like pattern. But what exactly determines this stripe-like pattern? So the answer is modular enhancers. So just to give you a refresher, enhancers are part of the DNA sequence which are cis elements which are present on the same patch of DNA as our coding gene, and which onto which different transcription factors can bind and cause transcription of the gene. And one important thing about enhancers is that they are modular, meaning you can essentially pick and choose whatever enhancer you want and put it on top of another gene, and they can function. So even though one of the enhancer is not functioning, the other can function. So they are modular. You can essentially just put them into their place and they will start working, and put them out of the place and their function will go away. So they are modular. So how does the stripe pattern of the different pair rule genes work with the help of modular enhancers? So this is where our coding gene starts, and upstream and downstream we have different enhancers for different stripes. So if a transcription factors goes down and binds to a particular enhancer for a stripe, so say stripe 3 enhancer, then in stripe 3 in that particular region only the particular gene will be expressed. And who are these transcription factors? These are the gap genes. The hunchback, the giant, the nerves, the cripple, their concentration determines which particular stripe enhancer will be active, because they are all competing for these enhancers. So wherever there is the optimum concentration of a particular gap gene, only there the particular pairwise pair rule gene will be expressed. And that is why the expression pattern of the gap genes is so important, because essentially they start the expression of the next round of pair rule genes. So this has been shown, the enhancer modularity. What they have done is that they have taken the enhancers, so say the stripe 1 enhancer, and added lag z to it. So lag z essentially will help us. So lag z, after you treat it for alkaline phosphatase staining, so it will give you a dark pattern. So essentially this will form a protein which will help us. What was the protein? Huh. Yes, lag z forms alkaline phosphate. No, it doesn't form that. So essentially lag z will mark the particular wherever this protein is being expressed, it will form the product of the protein, and when you put in the substrate for lag z, it will form a particular uh, pattern there. So essentially you can mark wherever lag z is being expressed. So when you add the stripe for one enhancer and followed by lag z, it is expressed right here, and then you do your lag z staining, and it will come up only in stripe 1. Similarly, they show that you have stripe 5 enhancer followed by lag z, and only stripe 5 expresses lag z. So essentially, these enhancers are why pair rule genes are expressed where they are. And when you combine two, say stripe 1 and stripe 5, you have LAGZ being expressed in stripe 1 and stripe 5. Now, interestingly, as you remember, I said whenever one of the gap genes is not there, the other gap gene which it opposed will fill up the entire region. Similarly, if in a giant deficient embryo you see that the expression, yes, Suba?
Yes, I sort of understood the picture. So it's a giant deficient embryo, and we see the stripe uh, 7 and 8, or what is the number of the stripe here?
It is 7 and 8 essentially. 7, 8 as well as 9. So essentially this entire
Okay,
region,
so they are all fused. But we also have the giant in the anterior
which is here.
part of the embryo.
Yeah. So
But
you're
they all,
asking why this band has not increased in this region. That's a really nice question. But if you look at it closely, you see that here there are changes here. You see this band is not as sharp as it is here. It is because
oh,
there are head. So this is an interesting idea that we will deal with in later part of the course, which is known as posterior prevalence, which means the gene which was here. So here
mm,
we had giant. So giant was making sure that only this region did not express whatever stripe 7 was expressing, and that is why we only had stripe 7 expressing this in this fashion. But when you remove giant from the anterior region right here, then what happens is that there are this stripe is from the this stripe. So is only being not only being formed by the concentration of giant being here, but also a anterior gap gene which is for causing the concentration to be correct here. But you can see the stripe is not as sharp as in the wild type embryo. This is because
okay.
the you see that the boundary is actually being hedged in. If you had a
Mm-hmm.
separate pair rule gene which was expressed in this part, so as we said that pair rule genes are expressed in alternate stripes. So this one and this one, the middle one does not express this gene. Similarly, there would be a pair rule gene right here whose expression pattern has increased, but you are not staining for it.
Okay.
Can you imagine? No, nahi jam raha. So how you can say this is that, say there was a staining for a different gene right here, like the
Mm-hmm.
example we had seen here, this example where we have two separate genes being stained at the same time. That is why we have an alternate one, like alternate expression pattern. Here we are not staining for two, we are only staining for one. So
Okay.
here this red part would have increased here all over this region because giant is not there. But since we have not stained for the red, we do not see it. If we had stained for the red gene, we would have seen expression being increased like this.
Okay.
Yeah. Okay. Okay. So gap genes are regulated by mutual antagonism among non-adjacent pairs of genes. Which of the following regulates pair rule genes? The answer is enhancer modules. Okay, moving on. Engrail gene is activated in the cells that will express hedgehog. High level of even skipped Fushitarazu and paired transcription factors control this activation. In an embryo deficient in Fushitarazu, only 7 bands of engrailed are seen. So these are essentially 3 different statements. You have to say either they are true or false. Yes, you are correct. It is X-gal staining. Yeah, that is what I was forgetting. It is X-gal staining, which so Laxi helps in formation of alkaline phosphatase, and the substrate is X-gal, which actually stains it. You're absolutely correct. Okay, so we have the answer true, which is the correct answer, but we'll let us look at it. So we are looking at how, yes, it is. Let us look at in detail. So the first thing that we'll look at is the next set of genes. So we have looked at gap genes, we have looked at pair rule genes. Next, we will look at segment polarity genes. So as we said, segment polarity genes are present at the so they are expressed at either the anterior or a posterior side of a segment. So we have engrailed right here, which is expressed at the posterior end of each segment, and we no engrailed right here, which is expressed at the anterior end of each parasegment, and wingless at the posterior end of each parasegment. You can see right here it is wingless it as the posterior end and engrailed at the anterior end of each parasegment. But how exactly are their expression patterns determined? They are determined again by the previous set of genes, which were pair rule genes. So we are looking again at the pair rule genes even paired, even skipped, and Fushitarazu. So a high expression of either Fushitarazu or even skipped give rise to engrailed. So wherever there is a high expression of even skipped, we have engrailed, or Fushitarazu, we have engrailed. Where we do not have either Fushitarazu or even skipped, we have wingless. So this is because this is how the diagram is drawn, but since these are pair rule genes, there will be another gene which is expressed right here. So it is expressed in this pattern. So one here, one here, one here, one here, and this uh, gene, which probably would be odd skipped, which is the other pair rule gene, actually helps in the expression of wingless. So high expression of even skipped or Fushitarazu, the pair rule genes help in the expression of engrailed in a spatially segmented pattern. So it is expressed only in the anterior portion of each parasegment, as you can see right here. And that is the first thing that we have to know. So pair rule genes, their pattern helps in the expression of segment polarity genes. Now in this particular example, we are going to look at how the segment polarity genes help in further morphogenesis. So say these two genes at one end of the entire segment were expressing engrailed. Now again, engrailed starts the expression of secreted signaling molecule, which is known as hedgehog. Hedgehog, what it does is that when it is secreted, it goes to the adjacent cell and asks it to start expressing wingless, which then diffuses to the other cells. In When it goes to an engrailed expressing cell, it asks it to produce more and more hedgehog. So this feedback loop determines that the cell at the posterior end will only express wingless, and the cell at the anterior end will only express engrailed, and these in turn cause morphogenesis in the entire segment. Those segment polarity genes are, that is why, so important. So this is what it shows here. So this is the question: the engrailed gene is activated, will express hedgehog. That is true. High levels of even skipped Fushitarazu and paired transcription factors control this activation, which is also true. Even skipped and Fushitarazu, wherever that is high, there is only engrailed expression. And an embryo deficient in Fushitarazu only has 7 binds of engrailed are seen, which is also true. So this is this entire question was statements were true. Is this fine? Yes, Suba?
What I'm not getting is even from the lecture that I listened, is that you know uh, so here, um, there is this different types of so this is after cellularization.
This is after
Uh,
cellularization. This is a very late stage embryo where all the segments are
okay,
also formed.
okay, all the segments are formed.
Yes.
So exactly where this is happening, and why this wingless expression and this engrailed expression are happening in uh, you know in they are diffusing. So
Mm-hmm.
the two cells are producing and they are diffused. So at what interval it is diffusing? So
Okay.
how many? So from one cell, how many cells fate is decided?
Okay. So let us go slowly. Till
Okay.
now, whenever we have been talking about, so after the gap genes, when we are talking about gap genes, these genes are transcription factors. So when they are expressed, these genes go around and help in expression of the pair rule genes. Till then, cellularization has not begun. To till this stage, when we see the stripes, the whatever one nuclei is expressing can go ahead and directly influence the other, because these are transcription factors. Most of the time they only hang around the nucleus, but since the cytoplasm is joined between all of them, a particular gap gene can go ahead and influence the expression pattern of a gene on the side. Yes, Suba?
Sorry, I also have one doubt. So at this point, there
Mm-hmm.
is yes, there is no cellularization. Only in the segment,
Yes.
the the
Here we
segment.
talk about cellularization. Yes.
So there only the cellularization happens where where all these nucleus are located in the cortex region.
No, 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 no. Okay. So when I say, so the nuclei being going to the cortex happens really
Hmm.
early on. So it happens all the way. Where did it go? Where is this image that I need? It is not here. Yes. So first, first thing that happens, becoid and nanos being on two ends, this is in
Hmm.
the oocyte. No fertilization has occurred. So you have one nucleus, huge nucleus being here and doing nothing. Fertilization occurs and the cells start, the nuclei start dividing. And this is the stage where the gap genes expression starts. The nuclei divide and they all of them go to the surface and stay at the cortex. So nuclei, 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 but the entire thing is one cytoplasm. So whatever one particular nuclei is transcribing can go and affect the one next to it, because essentially there are lot of nuclei. This is just the cortical arrangement of nuclei in a Drosophila embryo. Even
Okay.
at the pair rules gene stage, we still have
Yes.
nuclei all around the cortex. The boundaries have still not formed. So the pair rule genes, them being transcription factors, which normally only do stuff in the nucleus, because the cytoplasm is linked. So pair rule genes from one particular stripe go ahead and affect the other one right here.
Okay.
Fine.
Okay.
Now, by the time we go to the segment polarity genes, here cellularization has already occurred.
Okay.
So this is when, what do I mean by cellularization? It is the cytoplasmic boundaries being divided. So now the nuclear,
Okay.
the cells are now completely formed. So now the transcription factor from one cell cannot go ahead and do stuff in the other, but because there is an entire cell boundary in the middle. Now these cells have to start expressing ligands to go and do normal juxtacrine or paracrine signaling, because now the cells are divided. There is no longer one particular nuclei can directly affect the neighboring one. It has to go undergo transcription of particular secreted stuff, and that will do things, which is where we are at the segment polarity stage. Here is where we talk about
Okay.
cell signaling. Till here it is just transcription factors just going ahead and doing stuff to the other one.
Okay.
Understood? Now let us go
Yes.
and look at it in this light. Okay, so coming here, till this point when we had even skip, so the pair rule genes, even skip, fushitarazu, these are all transcription factors being expressed by particular nuclei, and these in turn activate the segment polarity genes. Once the segment polarity genes are activated, so engrailed and wingless both, so engrailed is a transcription factor being activated, and now cellularization has occurred. So now you see actually we make the cell boundaries. Till now we were not talking about cell boundaries. We were talking about transcription factors going and affecting directly.
Okay.
So now when the cell
Okay.
boundaries are formed, if the cells have to communicate with each other, they have to secrete ligands and undergo cell-cell communication instead of just expressing the transcription factor and sending it to the other nucleus. So at this point, we have already determined that which cell will be engrailed expressing, which will be wingless expressing, which is done
Okay.
at a previous stage when the nuclear the it was still a syncytium, not a cellularized embryo. Once this is determined, engrailed expressing cells, and engrailed is a transcription factors, it goes ahead and causes expression of the ligand hedgehog. Hedgehog now has to go all the way through the extracellular matrix to the other cell because cell boundaries are formed, no longer direct communication. Now it will go and ask the other cell to start expressing more and more of wingless, which is also it is also a secreted ligand. It will go ahead and diffuse to other cell and start asking them to express different stuff.
Okay.
You understand? So this
Okay.
is where we change from a syncytium to a cellularized embryo, because till here it
Okay.
was all transcription factors being expressed and diffusing and doing things. Here now we are going to start talk about cell-cell interactions via ligands.
So it's like the whole embryo cellularized.
Yes,
Uh, Okay.
yes, yes. Till
Okay.
this point, it was a syncytium in which there was one huge
That's
soup of cytoplasm where any protein one particular nuclei is expressing can go ahead and do stuff to the other. But here now
yeah.
it is going to be morphogens. It is still, these molecules are still called morphogens, but these are secreted molecules which will bind to a ligand and start a signaling cascade like the BMP signaling or the RTK signalings or the Wnt beta
Mm.
catenin signalings that we have already discussed. This is where we
Yes.
come in. Okay?
Okay.
Yeah. Okay. So engrailed gene is activated in cells that will express hedgehog. That is true. High levels of even skipped Fushitarazu and paired transcription factors control this activation. And in an embryo deficient in Fushitarazu, only 7 bands of engrailed are seen. All of them are true. Okay. Moving on. This also depends on the previous thing that we just saw. What receptor binds hedgehog ligands in the segment polarity gene model? Like, what does hedgehog bind? Any idea? Okay, so let us look at it. Uh, Hedgehog, the receptor for hedgehog is known as patched. Patched is the one that attaches with smoothen, and then we have CI, and then further and further expression. Wingless attaches to a partner known or a receptor known as frizzled on the other cells. I think we have, yes, we do have this. So this is again just enlargement of essentially these two cells. One of them has started expressing engrailed because of the pair rule genes asking it to express engrailed even before cellularization had occurred. Now, this transcription of engrailed causes a transcription of hedgehog. Engrailed being a transcription factor activates hedgehog. Hedgehog is secreted outside. It binds to patched receptors on adjacent cells, starts the hedgehog signaling, which finally causes wingless expression in the adjacent cells. And wingless is also secreted out, and wingless goes and binds to frizzled on the first cell, and actually this is a feedback loop. Now the wingless signaling in this particular, which is the Wnt beta catenin signaling, causes more and more expression of engrailed. So this feedback essentially makes sure the one cell only expresses engrailed and the other cell only expresses wingless. So what receptor binds the hedgehog ligand in segment polarity gene model? The answer is patched. And now we are going to deal with our final set of genes, which are the homeotic genes, and one of them being the UBX gene. So what is the consequence of deleting the UBX gene in Drosophila? The options are: the third thoracic segment is absent, the second thoracic segment produces halteres instead of third, the third thoracic segment takes the identity of the second thoracic segment, or halteres in the third thoracic segment are converted to legs. What do you think is the correct option? See, the third thoracic segment takes the identity of second thoracic segment. You're absolutely correct. But let us look at it. So by this time we have done a lot of stuff, which is gap genes, pair rule genes, segment polarity genes. The gap genes and the pair rule genes, both of them come together. They being transcription factors, they go and start expression of the final round of genes, which are the homeotic genes. And these are the master regulators of the body plan. And interestingly, when they are present on the chromosome in the same arrangement from anterior to posterior. So for example, the anterior pedia complex, which essentially you can see labial, which patterns the head region, DFD, which patterns the thoracic region or the last head region, sexcomb reduced, which starts the T1 region or patterns the T1 region, anterior pediate cell, which patterns the second thoracic segment, and similarly the bithorax complex, ultrabithorax, patterns T3 followed by ABD1, ABDA or abdomen A, patterning A1 to A4, and finally ABDB patterning A6 to the N. They are actually present on the chromosome in the same order. So they are so left is essentially the 5' end, the right is the 3' end, and here they, on the embryo it is the anterior position in the posterior position. Interestingly, all of these genes, if you, as I said, if you take one of these genes and place it somewhere else, then it will start making that particular body part in a different region. So this was first found by when people started did the experiment to remove ultrabithorax here, which forms the T3 region, which should have formed just one leg. But when you do a mutation, then interestingly the T3 takes the same appearance as T2. This actually is the same thing that happens with our giant mutant that we saw way back in case of pair rule genes. Essentially, T2 genes start expressing all the way to T3. So this entire region becomes T2. Do you get the
Okay.
similarity?
Yes, yes, yes.
So it actually flows over, and this is what is known as posterior prevalence that I was talking about. Whichever
Okay.
gene is expressed on the posterior, so antennae is T2, T3 is ultrabithorax, ultrabithorax is expressed in the posterior. So ultrabithorax actually can overcome antenapedia being expressed here. Ultrabithorax goes away, anterior region expands. If I took,
Okay.
and there is the other example where antenapedia, which helps in the formation of feet, is taken from here and placed in an anterior region like the antenna region here. Then antenapedia takes over and forms legs right here in the head. So whichever gene is expressed in the posterior can take over and start expressing its own properties if it is there. If it is not there, then the anterior gene takes over and forms the region it wants to form. So ultrabithorax not being there, the entire thing will be antenapedia. So T2. If ultrabithorax is
Okay.
expressed here, antenapedia is kicked out and only T3 will be expressed here.
Yeah.
This is known as
Okay.
posterior prevalence. So here is the ultrabithorax fly. So T2, which is should have formed a wing and a pair of legs. Now you remove ultrabithorax, T3 goes away. So both the regions look like T2. So you have four set of wings instead of two, because now this T3 region, which should have only formed legs, also forms wings. So you have four set of wings. So what is the consequence of deleting the ultrabithorax gene in Drosophila? The third thoracic segment takes the identity of the second thoracic segment. Okay? Okay, so that is all the questions done for today. Do you have any doubts regarding any of them? Then we can discuss them a bit further and try to figure out if you have anything.
No, for now I think it's good, but I have to read and I have to read and I'll come back with the question next session.
Yes, that will be great. So if you go ahead, and I would suggest Scott
But
Gilbert.
before that, there is one more question because when you spoke about the posterior privilege, so you
Prevalence,
mentioned that when
prevalence. It's
prevalent.
prevalence, yes.
Uh, so when so even in the giant mutation, where so even there was the posterior
Prevalence. So
yeah.
here,
So even okay.
so whenever we talk about posterior prevalence, we talk about homeotic genes or Hox genes most, because this is seen
Okay.
much better in Hox genes. Because as you saw that in this embryo, these are nuclei in the same cytoplasmic soup. So you can't take
Okay.
something here and put it here because it will diffuse all the way. So here posterior prevalence, even though I am drawing an analogy here, the reason why this expansion of the domain occurs is that all these enhancers, it is not that only one particular gap gene product will bind here. So you can't say, okay, stripe 1 only giant will go and bind, or stripe 2 only triple will go and bind. These gap genes are transcriptional regulators. They always compete with each other, and for some particular genes, for example, if this was Fushutarazu, maybe Krupel is an inhibitor and giant is an activator, and they actually go for the same DNA segment. So this enhancer can bind both of them. It is just if there is more giant, then it will outcompete Krupel and bind here and start expression. And if there is more Krupel, it will go ahead and bind here and start expression. Now, when there is no giant around, it cannot compete, because this is the same enhancer where giant was also binding, but here giant's expression was more because there was more giant here. Because you remember the gap gene pattern, giant was more here. So
Yes. Yes.
when giant was more here, it was going and binding to the same enhancer and making sure Fushitorazu is not expressed here. But once giant is out of the picture, now Krupal will go and bind to everything and increase the
Yeah.
domain of expression.
Okay. Okay.
That makes sense?
Yeah. Makes
And,
sense.
and
Yeah.
here I said that yes, the expression pattern does seem different, but you have to understand
Yeah.
that stripe 1 enhancer is different, going to be different from stripe 7 enhancer, if I can find it wherever. Yeah, or stripe 5 enhancer. So this same enhancer can bind giant, but it maybe activates the expression in this point. Or
Okay.
because the, as we said, that the enhancer are modular, so they can have different functions. So the same, if stripe 1 enhancer was there and giant binds, maybe in this case it activates the expression instead of down regulating it.
Okay.
It is a bit confusing, I understand.
Now I have to, I will read the book.
Yeah.
I will go through the lectures again and the notes again. Then, uh, and I'll come with questions the next class.
Yeah, perfect. Okay.
Yes, thank you. Thank you for your time.
Okay,
Have a
so
nice day.
yeah. Okay, thank you for joining. I will see you all next week. Thank you.
Yeah. Thank you.